Hello, I'm from UC Berkeley. My name is Oge Okpala, and over the summer, I worked in the Margaret Frank lab as well as in the Bruce Lewinstein lab. And I had almost two projects, one having to do with plant biology and another one having to do with like a more social science side. And so I worked as both a plant biologist and a social scientist. So as a social scientist, I was really excited to look into this research approach called community-based participatory research, or CBPR. And it's basically community members identify an issue that's going on in the community and they bring that up to scientists. And the two parties work together to address that issue, investigate it, and hopefully come out to a solution. And this works to graft knowledge, two different types of knowledges together, or combine two different types of knowledges, with one being the local expertise of community members and another being the scientific knowledge of scientists to sort of arrive at this investigation or experimental design. So I wanted to see if it could be adapted to a plant science context from more of its origins as a public health sort of practice. So I talked to Ithaca farmers, I talked to Cornell interns here this summer, and I also talked to members in my lab to identify barriers that are specific to plant biology, as well as propose potential solutions. So I have some barriers identified here, but I wanted to highlight funding opportunities. And we found that funding typically prioritizes the type of research that is done based on what it requires in these sort of funding grants. So we thought that there needs to be better infrastructure included in research institutions and funding organizations to incentivize CBPR and similar public engagement practices by both providing incentives and support. And then on the plant science side, we looked at sort of establishing a timeline for a graft junction to heal, because that's currently unknown. And in order to measure that, you want to look at secondary plasmodesmata, or these channels that open up between the two different parts of the plant, the bottom being the rootstock and the top being the scion. And this allows for cell communication as well as symplastic transport of nutrients and other things that keeps the plant functioning. And we wanted to take sort of these confocal or confocal microscopy images of the plasmodesmata as the plant heals. So we could take from that the density of plasmodesmata that develops during this process. We weren't able to get to the grafting, but we were able to identify that the two fluorescent protein lines that we used for this project were present in the two different plant lines. So we have TD tomato and GFP. And then as a negative control, we wanted to use these inducible lines or inducible callose lines. So you apply estradiol to the external portion of the plant, and that increases the deposition of this protein called callose that effectively blocks off the plasmodesmata. And this helps us investigate the role of plasmodesmata in facilitating both transport and communication. So we were able to identify that between an induced leaf and a control leaf, there was more deposition for an induced leaf versus a control leaf. We didn't have time to do all the computational analysis, but just from observing that, we were able to determine that. So in the future, we wanted to take these, or once we genotype the GFP line, we wanted to graft them together and sort of establish a timeline for like in 6-hour intervals at which there's or how much average PD density there is at each interval. And then once we have that, using the ICALS lines as a negative control, since they effectively block the plasmodesmata, we wanted to match the plasmodesmata density to the corresponding signal strain used in cell communication. So for instance, 25% restoration of plasmodesmata compared to an ungrafted plant, how intense is the signaling at that sort of point? So that's for the future. And I created a metaphor to sort of tie the two projects together. So we have grafting the scion and the rootstock together, the scion being like scientific knowledge. In CBPR, you combine that with local knowledge to create this sort of composite knowledge as well as enhanced public engagement. And then we have the plasmodesmata functioning as incentives, since incentives open up opportunities to practice CBPR. Plasmodesmata opens up sort of this communication pathway for between cells. And then we have barriers to implementing CBPR being like callose, which effectively closes the plasmodesmata. And then we have the symplastic pathway being support between the two parties to sort of facilitate this research approach. So yeah, that's my project in a nutshell.
I love your metaphor. That's amazing. I love when people turn science into life lessons. This is such fantastic research that you've been doing. Is this something that you want to continue doing throughout your scientific career? And as you move forward with your careers, integrating kind of scientific knowledge and expertise with like these kind of more big picture community issues?
Yeah, so my background is in integrative biology and media studies. So this project was a really cool way to sort of incorporate both, I guess, disciplines. And this is something I definitely want to pursue, finding ways, especially with science communication, to sort of facilitate and foster collaboration between both scientists and the general public. I think I was inspired from COVID. I feel like there's a very big chasm between the public and scientists. So I think that like a great way to bridge that gap is to get the two to work together to like address a problem and sort of establish that relationship of trust, as well as establish a relationship where community members feel empowered and comfortable coming to scientists with those sort of issues. Yeah.
Oh, that's so amazing. I love that. I think COVID was a big learning lesson for a lot of people. Yeah. And now for the folks at home who aren't too familiar with grafting, can you go into a little bit of detail? Because I find grafting just so exciting and I want everyone to be in love with it.
Yeah. Sorry, for the sake of time, I didn't really touch on it. But yeah, grafting is sort of, you take two different parts of a plant and combine them. And usually like for agricultural purposes, there's two different parts of a plant. So for instance, the scion being the upper anatomy, the upper stem and upper structures, as well as the rootstock being the lower anatomy, lower stem and rootstock. They both have their own sort of optimal traits that when combined create like a composite plant that has an even more enhanced performance compared to that of an ungrafted plant. So yeah, that's sort of that idea of grafting plants at least.
Wow, well, great work this summer. I hope that you continue integrating all of this important knowledge together. As a scientist, I'm so excited to see how our community changes. Thank you so much for sharing.